One EBC, you can go to File, Fetch by ID, PDB, one EBC, and you should get something like this. I'm gonna go and repeat what I usually do, which is go to the interactive preset number 1, activate it, and do the same for publication 1, so I can get this image that has a little bit more contrast so that you can see what I'm doing. That's easy to do, right? No problems there. You don't have to do it. I usually like this because the background in black only works if you have a very bright screen. If your screen is dim, or in the case of the projector, it's pretty difficult to see. So here we are. You already know how to get the sequence, right? We could go to Sequence in Tools, and from the list of chains, we can select the sequence we want. Yeah, no problem. That one appears after I select Tools, Sequence, and that window appears. Is that it? Yeah? Okay. Don't worry. Of course, I'm here for this. Now, we have A, B, which should be alpha and betas. A and B, chain A and B here, should be alphas. C and D should be betas. And this A for one EBC should be the only myoglobin that is there. So to make things easy, click only on one EBC. And then click on Show. We should get a window like that. Remember what I said last class? If you have a red rectangle or a red highlight, it means that even though the sequence should contain these residues, those are absent. In the case of this myoglobin, all of the residues seem to be present. Yes? If you click on File and File, Fetch by ID, you just need the code 1EBC in the PDB section and you get the structure. 1E as in Ernest, B as in boat, and C as in cat. And you should get something like that. Emily, you get?
It's just in my computer, but I have the crisis and opened lots of.
There are masks in yellow and orange.
Yeah, that's a good question. I was about to tell you. Notice the proteins are all of them alpha helical. And in the sequence, all of the regions that form the helices are highlighted in yellow. The protein, the hemoglobin, and myoglobin are alpha helical proteins. And the helices are highlighted in yellow. So this sequence over here from E, G, E, all the way to K and S, it's a long alpha helix. Yes?
Es que no, o sea, no entendí bien y le di clic y solamente me apareció una de esas ventanitas, pero no coincide con la.
Okay, so close it and do it again. Tools, Sequence, and you need to select the last one. Just the last one. So there's few reasons for you not to get the same window, and it could be that you don't have the same structure. It has to be named 1EBC. And what can you tell me?
Es que en la última, en lugar de aparecerme nada más ¿qué? Me aparece antes una que. De hecho, a mí me parecen los dos. Ah, no, es que la aparece.
Right, but because you didn't do what I said. No, don't lie. Look at this. Tools, sequence, you click the second, not the first one. Yes, and I said the first one.
There's file.
So the only difference you made is that you click this one, not the first one. Okay? So click the first one and you'll get the same sequence. Anybody else? No? Now, as your classmate asked, if you click on these yellow regions, you select only the parts of the protein that correspond to that alpha helical structure. Okay? If you click on the yellow rectangles, you select everything that has that property, and that yellow rectangle property means alpha helical. There's several ways. The easiest is select, clear selection. Okay, but still, what I want to show you is something to do with the sequence. Now we have this sequence, which is the myoglobin. There's only one chain because that protein is a monomer, okay? Now what we need to do is to go to Edit, Add Sequence, okay? Everything except, why did you click on everything? Just the last sequence. Close all the windows except this one. Now go again to Tools, Sequence, and select Sequence. Wait. And once you get to Sequence, Sequence. Just click on the last one. I know you did that, so just click on the last one. The good thing about this is you can close all of the windows and start over. It doesn't take much effort. Now, again, in Edit, on the sequence window, go to Add Sequence. And if you click on that one, it's going to open another big window that allows us to do many things. We could add a sequence from main text, from a structure, from a file, or from UniProt. And what I want you to try is to go to structure. Why? Because already we have a hemoglobin structure and myoglobin structure, so we don't need to look for other things. Now click here and select 5WOG-A, which should be alpha hemoglobin. And then click on Apply, not on OK, on Apply. And it should add that sequence to the same window. Now, still on this window, for adding sequences, click on the menu and select 5WOG chain C. And if we are correct after pressing Apply, and now we can close this window,
Is this an alignment?
the alignment should have the three sequences we are working with. The first one should be the one for myoglobin, the second one should be the alpha hemoglobin, and the third one should be the beta hemoglobin.
And how do we delete sequences?
In the same menu, if you really wanted to delete sequences, you go to Edit, Delete Sequences, and there is the list of the sequences you have there. Now these sequences should have been automatically aligned, but let's assume they weren't. If you click on Edit, there is another option, Realign Sequences. Let's go to that one. Click on Realign. We get this window over here. In my computer, the default is Muscle, and it's gonna update the current window. I usually change this maximum number of iterations to 160, but that is optional, you don't have to. And click OK.
Can we click on apply?
You can click on apply on OK, doesn't matter. It shouldn't change a lot.
Pero igual ponemos en
Yes, realign again. Just click OK.
pero en program
Y acá arriba.
o le dejamos en
I would recommend muscle,
currently.
but that's my personal opinion. Say that again. The same window, realign sequence, same window. So here is my window again, realign. Mine says the current alignment window, so that it doesn't open another window. And muscle for the program, 160 iterations. Those are my personal settings. Any should work anyway. What's that? So open in edit the realign sequences window, put this data on the window and click OK. That's all you need to do. But this already happened automatically. I'm just redoing it so you can know how to do it yourselves. Yes. And just click OK. This already happened before and we did it again. Now to see the results, you go to headers and select one of these three options: charge variation, consensus, and conservation. To make this easier, let's start with consensus. That is going to be similar to what we already saw in tea coffee. Now we have this other line of consensus which show us in red, in capital letters, residues that are identical. In lowercase in black, residues that are conserved. That is, they are not identical, but they are represented often. Look at this one, for example. In this position, there's a lowercase s, a serine, and in the three sequences, we have two serines and a threonine. Threonine and serine are amino acids that have a hydroxyl group, so they are conserved. Over here, we have a gap because there's a valine, an alanine, and the other one, there's a gap. So that is a non-conserved position. In general, we can see that these three proteins have very few residues that are conserved. Since we have the structures, we can actually click, for example, on this histidine, number 61, 2, 3, 4, 65, and select across all of the sequences. What should have happened is that back here in the structure, now we should be able to see which amino acids those are. And it turns out that it's one of the residues that it's at the heme group, in the case of hemoglobin, but it's also one that it's bound or near the heme group in the myoglobin. Yes?
Can you put
The sequence alignment? There you go. What's that? They should be visible already. What I'm doing is that by selecting, I highlight them and can find them easily, or fairly easily.
Teacher, how can I move myself, well, the camera in the X and Y axis, not just rotating?
Oh, that's a good question. Let me get over there. It's supposed to be with the shift key, control, oh, that's selection, then, oh yeah, it's okay. So if you press the right click, zoom only. The middle one, translate, and the left, rotate.
Rotate, okay.
Is it free?
I cannot see it. Oh yeah, well you can see it.
Yeah.
Okay, so this is part of the illustration I'm trying to make. Whereas in the sequences they are already aligned, the structures are not. So that makes it harder for us to look at these features. I'm going to show you how to align the structures. Here I come again. Now to align the structures, go back to the main window, tools, structure comparison, matchmaker. What are you laughing? What does that mean to you? Yeah? This is not Tinder, don't worry. Yeah, right. Grinder. I don't question your preferences. Doesn't matter. This is for structures. Okay? So are you don't know? Then I'm not supposed to tell you.
I'm talking about Mulan.
Doesn't matter.
Disney, the
Doesn't matter.
matchmaker.
Yeah. Let's not go into details about your pop culture. Are you there? Have you opened the window? What does the window offers as options?
It says like structures to match. Is it reference?
But what is different from the sequence, it's not telling us which chains.
It says, do we further restrict matching to curves?
I'll show you which is the one I will pick. But is everybody there in the same window?
Yeah.
You sure? Okay. So because this one doesn't give us the option of a specific chain, what we want to is to click on select a specific chain in reference. And this time, what we want to do is select chain A, the one that is the alpha, and on the second part of the window, the myoglobin, like that. So what this program is going to do, or this window is going to do, is going to take the myoglobin and the alpha hemoglobin, keeping the alpha hemoglobin fixed in space, and the myoglobin is going to be moved to the other position. And they are going to be to try to be superposed the best possible. Well, a juxtaposition doesn't work here. It's actually a superposition based on the alpha carbons of the amino acids. So it's a restrained juxtaposition, if you will. And it usually works very well. So don't change anything else. Keep clicking OK. And what you should see should be pretty much instantaneous. And now, because of the selection, we should be able to focus on the region that it's important. So two proteins that are very different in sequence, they have a similar structure and a similar function. They look a lot alike when you compare the structures. This is similar to the molecular biology central dogma that says that DNA has to become mRNA and to become protein. This, for biology, for biochemistry, is a similar dogma, that form determines function. Because you are rotating about a center that is not in the protein. Is there anything selected? So go to actions and there you go. No, you can deselect and then set the focus. And try to rotate now. Now we know how to obtain the identity of the sequences. For structures, there is no such thing as an identity, but there is a value that sometimes is useful. Here in favorites, if you go to reply log, we get this window. And this window says there's 104 pairs, which refers to alpha carbons that are compared. And the distance between them is about 1.1 Armstrong. This window, you can open it in favorites. It's called reply log. Favorites, reply log. What? It shouldn't be exactly the same, but it should be very similar.
Yeah, it is.
So here it says that of 104 atoms that were compared, the distance between them is 1.1 Armstrong. If 133 are compared, the distance is 2 Armstrongs. These numbers are difficult to bring into context, but if they were identical or very similar, usually 0.5 is what you expect. Between 1 and 2, you have some differences. Sadly, these numbers are like that. They are not going to be dramatic. They are not going to be 10. They are not going to be 0. They are going to be between 0.5 and 3. But still, it gives us something to report as a quantitative function, or a number, if you will. If we go back to the alignment sequence, the sequence window, we can also get a percent identity. Remember that we got that in tea coffee? This is the window that from this alignment can calculate the percent identity. But in contrast to what we did in the test, it's not going to be only for 2 sequences. It's going to be for the 3 sequences. But these values are going to be pairwise. That is, one sequence compared to the another, and the first one to the third. So select in this window the longer sequence length
Info?
and compute. So info,
Info?
percentage identity, and here the longer sequence length.
Longer sequence. 5.
And click OK. You can close the window. And here it goes, the list of all of the pairs that can be made. A versus ABC, A versus itself, 100%, A versus C, 41%, C versus 1ABC, and so and so. Because here we are comparing A and C, which will be alpha and beta, this should be very similar to the value that you reported in your test when you got the beta subunit in the test. Some got delta and somebody got gamma, but you should have very similar values. So why do I like Chimera so much? Because it lets you do this. Align sequences, align structures, and compare the information from known sequences to the known structures. And we're not doing bad in time here. Okay, now, what am I, if I were to ask you, what else should I show you how to do? What would be the first thing that you would think of? No? What about saving your work? One thing that I already shown you is that if you go to File and save the session, you save everything that you just did. That is the first save that we have. But because these windows work independently, you can save the content of the reply log independently of the sequence alignment or the structure alignment. So on reply log, you have this prominent save button. If you want to use this data, you can just click save and save your log file. And go ahead, do it. This one is going to be the simplest. If you click save here and select, let's call it hemoglobin.txt and save. You can name it whatever you want. Here, I name it hemoglobin. Yeah, you can name it. And that file is going to be like this, a text file. You can open it anywhere you want. If you are doing some analysis and you really want the data, don't forget to save that data. I'm going to close it. If I was to save my sequence alignment, I could do it in this window. In the sequence window, I could select Save As, and then select a format for my alignment, and save the alignment too. In general, I think that the FASTA format or the Clostal are the most common, So I recommend that if you perform a sequence alignment and want to save it, pick one of those two. I usually select FASTA just because it's the most common. Sometimes I save it both in FASTA and Clostal. Clostal is easier to read. If you are reading it yourself as a human, Clostal is easier. So save as, I'm gonna save, how will I call it? Alpha, hemoglobin alpha, hemoglobin beta, and myoglobin. Dot FASTA. And notice, if I double click on FASTA, it recognizes a file that Chimera should open, and this is the file I saved. I'm gonna close that. You can open it also as a simple common text file. Oh, I don't have that file open. Oh, come on. So here is an example of how the FASTA format looks like. It's 3 independent sequences, but with the gaps of the missing sequences in the alignment. Just by looking at this, you can get the idea that roughly myoglobin is slightly longer than hemoglobin.
Yeah, teacher, we can open it in our computers.
You can.
No.
Yes, of course you can.
We have to.
So what I usually like to take bets when people say things cannot be done.
I mean,
What are you offering?
it says that. How do you want to open this file?
And how do you want to open that file? Use any text editor. Note edit, write, even Word if you want. Just try it. Don't believe in me. If you cannot do
Not
it,
bad.
nobody can do it. Try it. And tell us what happened, because only the people behind you can see the screen. So you managed to open it?
Okay, teacher, I don't like it.
I know, but I don't mind. Now I'm gonna save the file again in Clostal, just so that you see. File, save as, Clostal. Wow, what should I? ALN, that's a good, decent name. Oh, come on. Oops, what the?
Teacher, and with all of this, what do machine learning scientists do?
Anything they can.
Like what?
Not much.
Not much?
No, machine learning is just pattern recognition, which means that if you make a machine to do it for you, it's just do it with tons of data very fast. But they don't. So it's like a cookie cutter thing, that it's gonna be able to recognize things, but it cannot give you insight into mechanisms.
But what are they trying to discover?
Nothing, not really. Accelerating things is what I think they are doing. So this is the Clostal alignment, and as you can see, it includes the nomenclature we already know about identity for the asterisk, very well conserved, the colons, dots for conserved amino acids, and nothing for sequences that are very different. So this one for humans is very easy to read. But as we have seen, for example, in tea coffee, you need FASTA to perform further analysis. Questions? No? Okay, so how to save that information. Now, in order to make something nice out of this, I'm going to open my side view panel, go to this lightning thing, and usually trying to print an image like this, it's actually very difficult because in paper you lose all kinds of perspective. So what I usually do is here in lightning, in the side view, go to shininess and reduce that reflectivity to 0. That makes a little bit nicer images. Can you find that? What's that? Well, don't let it sunburn. What did you click? Are you sure you click on shininess or did you click on illumination? Where is brightness? Ah, well then, whatever you did, undo it. An easy way to undo mistakes here is to go to the presets and click on them again. So go to this window, select in this menu shininess,
Side view.
and change the reflectivity to 0. That's all you have to do.
But like from oh, okay. So we go to light. Light.
Yes, it has to be lightning.
Yeah.
And it has to be then shininess.
Contrast.
No, shininess, reflectivity.
Ah, okay, yeah.
Now in effects, click deep queuing. And then in the tab for side view, pick the yellow line that is behind the protein and move it closer to the center of the protein. You just click on the line and drag it. And what you do with that line is that some things that are beyond that line are kind of hidden. What? You just click on the line, on the yellow one, and drag it closer to the center of the protein. And once that's done, now you can go to File, and you can save a very nice image. This is the menu for saving image. Save image. You can select TIFF, PNG, JPEG. I usually go for TIFF. And my only, by the way, this screen might be suited to my own preferences. The only thing I do recommend you do is to change the super sample to 4x4. And that's it. Give it a name. It could be image. And if you click save,
Is TIFF vectorial?
actually I don't know.
No, it's raster.
Yeah, I don't know this version, but you can save in both ways. I don't know in this particular one which is it. But if you want raster, or if you prefer vectorial, this is not the best option. For raster, EPS it's better, but. And here is the TIFF. Which one looks better? Here we go, side by side. It even looks nicer on the right, doesn't it? I don't know if you are suffering in pain or. So the easiest thing, so if you have this problem where you cannot locate where the structure is, it disappears, or you cannot really control it, my best advice is clear the selection, then then go straight into actions, focus. And that is going to reset the view, and you can restart what you were doing.
Professor, I have a question.
Yes, that's good.
Why does my image have like como si estuvieran apuñalando. De lo que sea.
I really don't understand that, so I'm gonna have to go over there. And if you have opened again one of those websites, Emily,
Yo no puedo guardar mis And it happened with the other one too.
well, I gotta say, you just have to stop using Windows. So can you show me how you saved it?
Yeah.
One of the tenets of troubleshooting is actually know what you did. Otherwise, it's difficult. What you got there? What are you showing?
So I can see.
You can save it because you have only permissions to the folder of your user. So if you try to save it anywhere else than the user folder, Windows is going to tell you that you can.
And how
No, a ver, cierra.
Hang on.
¿Qué te salió?
Que no,
Where? So do you know any way? No, you probably. Go to users. Practical users. Come on, you. I know, but you can. You know the letters. ABC. Okay, go to desktop. Now, save it.
Ya, here. Sí, fusión 3, 4.
Ay, como si te
Sí.
Wait, don't.
Ah, fusión 4.
es que me parecía sin automático. O sea, tú también lo tienes.
¿Sí?
Wait.
No.
Okay, first, save it in a different format and save the file name without spaces.
And what format?
Just say it different. Go for PNG.
Sí.
Okay, and try it again.
Ah, sí, ya.
So it might be an artifact of the program or the file format in Windows.
But why didn't it happen?
Oh, it's Windows, come on.
Pero it's there. No, it still has it.
Yeah, but look at your image.
Mira, Elba, ya no me dijo nada.
You can just crop it here and that's
Pues
it.
es que me dice Lau, digo, dile a Elba. Y le digo,
Elba.
Me mandan esta. Aquí.
Okay, now, did everybody here manage to save an image? Yeah, you sound disappointed.
no,
Okay, well, if you want to improve it, tinker with the settings. Now, let's do something slightly different. We compare, and kinda, I've been leading to this. In the last question, the extra question of the test, I asked you, what is different between just the sequence and the actual protein? And it's the heme group, right? So in this case, what we're gonna do is select everything that is not protein and remove everything that is protein. So in the select menu, go to residue, click on all standard amino acids. If you need glasses, Selva,
No.
really get a prescription.
No.
No? Okay. You can always sit closer to the screen. That is cheaper.
no,
No, she can?
no,
Okay.
she's with us.
Hey, if she is taking you as a slave, there's places you can complain.
Oh my God, teacher.
Okay, everybody done that?
I went on.
If you click on that, and now in actions,
Sí, tu cara va a cambiar, no me igual.
first atoms and bonds, and click on hide. Now only the atoms and bonds will disappear. You have to go to rebonds and hide them too. And now we have only the heme group. Now what we want is to select only, well, we don't have to select it. We can just drag our view to the heme group that is blue, which corresponds to the myoglobin. If you manage to get this far, now what we are comparing is just the heme group between these two different proteins, one from whale, one from human, and they are very similar. There's actually very few differences, this piece of the heme over here that has a different conformation. The metal, still the same, but the ligands are different. For human, it was crystallized with water, and this other molecule, I think it's cyanide. So this myoglobin was poisoned.
So it's like the little.
It is. Now, there's a very nice trick to make good images. If you wanted to save these, but you don't like them superposed as they are, you can make one invisible by unclicking the show square in the model panel, save this image, then make it visible and make invisible the other one and save the other image. And that way you can have a side-by-side comparison of these two heme groups aligned through the protein.
Where does that go?
Favorites? Model panel.
Bottom panel?
The first one. Model.
Panel. Which one do you want to disappear?
Whichever, doesn't matter, because you can make it disappear and reappear. You select, okay, I want to save the hemoglobin alpha first, I disappear the second. I want to disappear the myoglobin. I want to make only a picture of the myoglobin, destroy the other one.
And I change the color.
You can do pretty much whatever you want, yes. So, Chimera is a very powerful tool. As I told Paul just a second ago, it's free, but it has many, many more applications than what I showed you. This is just the general and was to illustrate, and I planned to illustrate that sequences and structures can be analyzed together. You can technically use this one just to perform sequence analysis, for example, or only structure, or many, many other things. But for the time being, I want to just cap it with what I showed you. Please check the videos if you want to repeat these things again, and click on the program, open files and click on them, do things. Experience is the best teacher, but for the next class, we're going to start working with a different, slightly different topic, so we're going to leave this behind. However, if you have questions or if you want to have a repeat of some other features of the program, we can have them. Hey, life is complicated. New things are easy. I don't think so, but then again, I'm biased. Easy to use? In what? Intuitive. Well, it's not intuitive because you are probably more used to things, Windows programs that have slightly different interface. Different interfaces. But again, experience is going to help you. Any other questions, guys? No? Then let's go out for the weekend. Have a good one.